Hi everyone, for this mini lecture we are going to talk about fat cells, specifically adipocytes, and how triglycerides are liberated. So that means how triglycerides are broken down into glycerol and fatty acids to generate energy. So let's begin by talking about this slide in adipocytes. Essentially, adipocytes are large empty vacuoles that fill and empty depending on dietary intake and energy output. New research confirms that fat cell numbers are maintained in adulthood. There has been varying schools of thought about this for a long time. Earlier evidence indicated that adipogenesis could continue if fat cell volume was increased to a certain point, but not everyone thought this was occurring. Regardless, new research techniques are supporting the idea that the number of fat cells stays constant in adulthood in lean and obese individuals, even after marked weight loss. This indicates that the number of adipocytes is set during childhood and adolescence and reinforces the importance of exercise and proper nutrition during childhood and adolescence, because the number of fat cells you have stays with you in adulthood. And what happens then is not hyperplasia, but rather hypertrophy of those fat cells, that they could increase in size or decrease in size depending on nutrition as well as physical activity. So this picture actually shows scarring from liposuction. So liposuction is the only way to actually remove fat cells from the body. But this picture illustrates how the removal of fat is an art, that once it's gone, it doesn't come back, and it doesn't redistribute itself either. Now let's talk about the storage of fats as triglycerides. So adult humans ingest approximately 60 to 150 grams of lipids per day in our diet, of which 90% of those are triglycerides. So we eat triglycerides, and then through the process of digestion, we're breaking down the triglycerides into 1 glycerol and 3 fatty acids. But following absorption, the products of lipid digestion are resynthesized into triglycerides and stored in adipocytes. So we break it down through digestion into the smallest form, and then we rebuild them back together, or we reassemble, and then we bring those to the adipose tissue for storage, and they'll be mobilized when we need to use them for energy. So remember, what type of functional groups are in triglycerides? This comes back to our first module on biomolecules. The answer here is esters, that we have 3 esters. How about fatty acids? What functional groups are in a fatty acid? And you should remember that these are carboxylic acids. Now let's discuss the release of triglycerides from adipose tissue when we need them for energy production. This figure highlights the 2 locations of storage of triglycerides. One is in the adipose tissue, where we have triglycerides stored from 50,000 to 100,000 kcals. That's why fat can provide a long-lasting form of energy. And then the second area of triglyceride storage is in the skeletal muscle. So in a trained skeletal muscle, we have intramuscular triglyceride storage, or IMTGs, of approximately 2,000 to 3,000 kilocals. So this makes fatty acid breakdown and use very proximal to the muscle. So the working muscle, when you're exercising, we don't have to take necessarily the blood plasma glycerol or free fatty acids. We instead can break down our intramuscular storage of fat, which makes it very proximal to the citric acid cycle and the mitochondria, where we see fat metabolism happening. So adipose tissue can release our fatty acids, free fatty acids and glycerol into the blood plasma. Remember that free fatty acids cannot travel by themselves. They need to have a taxi, and that taxi is the protein called albumin. So albumin is the taxi for free fatty acids to travel around the body. Free fatty acids can be taken up from the blood into the muscle, the working muscle, and as we can see, those fatty acids go into the muscle. So our concentration of fatty acids in the muscle comes from our IMTGs, as well as from free fatty acids within the blood plasma. Now let's discuss triglyceride mobilization. So how are triglycerides released from the adipose tissue as well as from the IMTG storage within the muscle? So first of all, there are two hormones that are very important in dictating TG mobilization, and these are epinephrine and glucagon. Okay, so we see at the top, we see epinephrine, which binds to its receptor on a cell membrane and basically catalyzes a cascade of events that leads to the release of an enzyme called hormone-sensitive lipase. Hormone-sensitive lipase is sensitive to the action of hormones, hence its name, primarily the one hormone being epinephrine. So when epinephrine is high in the blood, we see an increased amount of the activity of this enzyme called hormone-sensitive lipase, which catalyzes the breakdown of triglycerides, TAG, to diglycerides, DAG, and then from diglycerides, DAG, to monoglycerides, MAG, until we're eventually at the glycerol backbone. And each time we break down from a triglyceride to a diglyceride to a monoglyceride, we're releasing a fatty acid into the blood. And so this is how we break down a triglyceride. That essentially we need this enzyme called hormone-sensitive lipase to catalyze the conversion from a triglyceride to a diglyceride to a monoglyceride. And so that enzyme is very important, and it is catalyzed, or it is increased its activity through a hormone called epinephrine. As well, glucagon is another hormone that catalyzes the activity of hormone-sensitive lipase. Glucagon is released when blood glucose level is low. So it responds in opposite of insulin. Glucagon is released when blood glucose levels are lower than homeostatic control levels, meaning that we need to start liberating fatty acids and using fatty acids as an energy source to increase our blood glucose. And so these two hormones are very important in regulating fat metabolism. Epinephrine as well as glucagon, and they activate the enzyme known as hormone-sensitive lipase. Now let's talk about glycerol. What do we do with glycerol when it's free-flowing within the blood after we just broke down a triglyceride? Well, glycerol that is produced from the breakdown of triglycerides can have two possible fates. In the first, it can be used to make more triglycerides. This requires two steps. Firstly, it is phosphorylated by the enzyme glycerol kinase, which we see on the left of the screen. So we have glycerol, we add an ATP to it, and phosphorylate it to glycerol 3-phosphate using the enzyme glycerol kinase. This enzyme beautifully follows the rules of enzyme nomenclature that we learnt in class. The phosphorylation of glycerol forms glycerol 3-phosphate, which goes through a series of 3 additional steps, which is on the right, which you don't need to memorize. And this is able to remake or resynthesize triglycerides. Obviously, it has to react with fatty acids, but it is not as simple as that. The fatty acids are activated, which is something that you will understand at the completion of this module, but not right now. Okay, so when will this happen? Well, this storage of glycerol, or creating new fatty acids and new triglycerides, is favorable during a fed state. So when we have enough energy, we'll recreate triglycerides. So we'll use that glycerol to combine with available fatty acids that we don't need to produce energy right now, and we will store it back as triglycerides. So essentially, the process of breaking down a triglyceride and then resynthesizing a triglyceride is a reversible metabolic process. The second fate of glycerol is that it actually enters glycolysis and or gluconeogenesis at one common site. So the second fate is that glycerol can act as a fuel source. Firstly, it can be converted to DHAP, so dihydroxyacetone phosphate, which is in glycolysis. So if you look at glycolysis on the right-hand side, we can see the metabolic pathway, and that's circled in red is DHAP. And glycerol can be converted into DHAP at that step. And then it will enter glycolysis and do exactly what what the glycerol aldehyde 3-phosphate does. So it can act as an energy source. So it can enter glycolysis or gluconeogenesis to generate glucose. So the first step is really to convert glycerol into DHAP. So first of all, we have to phosphorylate glycerol to glycerol 3-phosphate. And then the second reaction is we need to dehydrogenate glycerol 3-phosphate. So you see NAD, the coenzyme there, and oxidizing glycerol 3-phosphate to form DHAP, dihydroxyacetone phosphate. And depending on the tissue needing energy, whether it's the liver versus another, DHAP will either go through glycolysis or gluconeogenesis to generate new glucose. Ultimately, the energy status of the cell will dictate the fate of glycerol. Glycerol will either be resynthesized with 3 fatty acids back to a triglyceride if we don't need the energy at the time, or glycerol will be converted to DHAP and enter glycolysis and form glycerol aldehyde 3-phosphate and follow the sequence of events of glucose oxidation. Now let's talk about what happens to free fatty acids once they are mobilized from triglycerides. Free fatty acids can move through the cell membrane of the adipocyte into the blood. They're lipid soluble, so can just pass through the cell membrane without the aid of any transport proteins. Because they're lipid soluble and not water soluble, they have to be carried in the blood, which is accomplished by the protein albumin. Once they reach the tissues, though, they can diffuse directly into the cell, again because they're lipid soluble, and can be metabolized for energy in a process which we're going to talk about a little bit later in more detail. However, the brain, the red blood cells, and the adrenal medulla can't use plasma-free fatty acids for fuel, so they need a source of glucose. We'll see later how glucose can be provided to these tissues even when glucose, glycogen, or fatty acids aren't used. For example, when someone's on a very low carbohydrate diet, there's preferential sparing of the small amount of glucose within the body for the brain, for the red blood cells that don't have mitochondria, and to the adrenal medulla.